Hello. We are going to be studying the homeostatic functions that occur in a cell. So prior to this lesson, we talked about the different homeostatic life functions. Homeostasis is maintaining a constant internal environment, and the key thing about homeostasis in a cell is that all of these different life functions are kind of happening simultaneously, and in many cases they rely on both to happen simultaneously and multiple of these to happen all together in order for any one thing to happen. So for example, respiration is the process of getting energy from food. Well, in order to get the energy from food, you're going to need to have sugar, and sugar is going to require the ingestion of food. So ingestion has to happen. It's going to require oxygen. Oxygen is going to require absorption. And then you're going to need to release the excess, the waste from that. So the liquid waste that is produced, water and CO2, that's going to be released by excretion. So just to carry out respiration, you need ingestion, absorption, and excretion to all happen. So this is a diagram that you all have, and for this diagram, we're going to identify the different parts that are shown here. So the first part right here, what I see is a larger food molecule entering this diagram and entering through this little pocket. This is going to be, set this up. This is going to be ingestion. I need to keep it like that. Sorry. That's going to be ingestion. The second step is where now the food is in this pocket and it's going to be now combined with these enzymes that are from the fused lysosome. So this was maltose. Now you have the enzyme maltase in there, and it's being broken up into individual glucoses. So this is digestion. So the next process that's going to happen is going to be process number 3, which is the old cell parts going to be released from a vacuole. This is going to be egestion. The fourth process is where I see oxygen entering the cell, and I have a lot of oxygen on the outside and a small amount coming in, so it's moving through by diffusion. This is going to be absorption. The fifth process over here happens at the mitochondria. It's where the glucose is entering in, oxygen's coming in, then CO2 waste and water waste are being removed with the whole purpose to produce ATP, which is energy. This is going to be respiration. The sixth process right here where I see the CO2 and water being released, that's going to be excretion. The seventh process right here, so in this is a picture of digestion, but instead of maltose being digested, it's going to be a ingested protein that's being digested, and the ingested protein is then going to be broken down into little amino acids, and then the amino acids are going to form into a long chain over here at the ribosomes. So this is going to be synthesis. The materials that are formed, a lot of times they're going to enter into the endoplasmic reticulum and then move around the cell through the endoplasmic reticulum. Moving around the cell is circulation. Some of the proteins are going to be encapsulated in a vesicle, travel through the Golgi complex, where they're going to be folded into a particular arrangement so that they can be secreted. Finally, inside the nucleus, you'll see the DNA copying itself, and also the centrioles over here getting ready to duplicate and organize the DNA, and that's going to be reproduction. So this next picture you're going to see is a color-coded picture of this. So it's using the same color code scheme that I had in the previous slide. So the reason why I have certain things colored the same thing is because they kind of have a similar function. So all the things that are green are processes where things are going across the cell membrane into the cell. So ingestion and absorption, both going into the cell. The two brown things are both processes where things are leaving the cell. So they both have to do with waste leaving the cell. The red processes, the ones that are outlined in red, those are going to be things where useful materials are being moved to where they need to be. So if they're being moved within the cell, that's circulation. If they're moving out of the cell, that's secretion. The yellow process there is digestion. The purple process is respiration. The pink process is reproduction. And then the blue process is synthesis. So again, the brown and the green ones are linked, and then the red outlined ones are linked. Again, the red outlined have to do with moving useful things. The brown ones are going to be moving waste products out, and then the green ones are going to be moving food or other useful materials in. And then the main difference is for ingestion. Ingestion is going to be large materials. Absorption is going to be small materials. Excretion is small materials leaving. Egestion is large materials leaving. For secretion, it's useful things leaving the cell. Circulation is useful things going to other areas of the cell. So I'm going to now shift to your note sheet here, and we're going to fill in this note sheet together. So ingestion and absorption, I put them together. Again, these were the things that were green, so it can add a green and circle them. These are the green processes over here, and the parts where they occur, this is going to be the cell membrane. Okay, so the other thing that we could put in here is going to be a vesicle. So the vesicle in ingestion is going to form a bubble around the food. Oh, sorry, I made a mistake on here. This would be the membrane forms a bubble around the food, which is going to be called a vesicle. Then absorption, the small materials go directly through the membrane. Okay, digestion is going to be our yellow one. No, I'm not going to write in yellow, but as it says, food is broken down into raw materials. The parts where this is going to happen in the vesicle, and it's going to happen with the aid of the lysosome. So the vesicle is going to store food. Now, sometimes that vesicle here, this might be called a food vacuole, just so that you know the other terminology. So the vesicle is going to store the food, and then the lysosome is going to inject enzymes that break up the food into its little component parts. Secretion and secretion. So this is the one we said we outlined it in red. So we'll write this one in red. So the raw materials go to where they're needed, inside circulation, outside secretion. So there's a bunch of parts that are involved in this. This is going to be the ER, the cytoplasm, the Golgi, vesicles, and membrane. So the ER are tubes that help transport substances in the cell. The blank carries materials to the blank, which modifies the product. So a vesicle is going to carry materials to the Golgi complex, which modifies the products for and packages them into vesicles for secretion. And then for secretion, then they're going to leave across the membrane. Synthesis is the one we had in, I believe we put that one in blue. So raw materials are put together to make usable substances. Now, the picture that we drew was a picture of an animal cell, but there's also plant cells, and plant cells do synthesis, a specific type of synthesis called photosynthesis, and they do that in the chloroplast. So we have the ribosome, the chloroplast. And then there's also going to be the nucleolus, which builds the ribosomes. So many amino acids are put together to make new proteins at the ribosomes. So excretion and egestion. So this is going to be, we'll highlight this one in, let's make it brown. Let's not worry about brown here. Let's just find a brown pen. There you go. So waste goes in and out. So this is going to be vacuoles and the membrane. So liquid waste leaves through the cell membrane. So it's going to pass directly through. Solid waste is carried in vacuoles. Okay, respiration. Respiration, I believe we did that one in purple. So we'll do this fancy color here. So what's the part that's going to have this? This is going to be the mitochondria. So the mitochondria, oops, mitochondria, it's disappearing on me. So it burns sugar with oxygen to make energy. Then reproduction. So reproduction, we will, oh, my second mitochondria was, all right, let's try that again. Mitochondria. All right, my last one is reproduction. So we can highlight, we'll use a pink highlighter here. Reproduction I made in pink. And for reproduction, there's two parts. There's the centriole, and then there is the chromosomes. So the chromosomes are going to store information that is needed in the new cell, and then the centrioles are going to separate the chromosomes and its copy. So that is ultimately what needs to happen in this process. So the next thing that you would talk about here is how might all these processes go together? So if I look at this is an example of a process here. And in this, I'm saying in order to secrete a usable protein, a cell requires multiple steps. So we want to describe how processes X, Y, and Z are needed to secrete a protein. So we identify process X is digestion, process Y is synthesis, and then Z is secretion. So ultimately, we want to get a secreted protein. So how do we get to the secreted protein from here? Well, we need an ingested protein first in a vesicle, and you need a lysosome to break that ingested protein into different amino acids. Okay, so think about it like if I started off with Legos and I had a Lego castle and I wanted to build a Lego rocket ship. Well, the first thing I need to do is break up my Lego castle into individual pieces. That's your digestion process. It happens in the vesicle. Then I need my plans and I need to put them together. So that happens at the ribosome. So now I'm building my Lego rocket ship. Then I'm going to package it up into a new package. So that's my Golgi complex. And then I'm going to give it to my friend, and that's going to be the secretion. So, in order to release a useful protein, there needs to be amino acids to build new protein. These come from the breakdown of an ingested protein, which happen at process X, digestion. This involves a vesicle and a lysosome fusing together to mix ingested protein with enzymes. The amino acids travel to the ribosome, where they are put together in process Y, synthesis. Finally, they are packaged and released by the Golgi body through the cell membrane in process Z. So I go over that for you for a reason, because what I'm going to do, and we're going to do this in a Nearpod activity, is I'm going to give you another scenario where you're going to get a similar picture to this. I'm going to choose 3 different processes, and you need to explain how those 3 different processes have to occur together in order for one of them, for the cell to carry out a specific function. Okay, so that's the conclusion to our cell homeostasis notes.